So thank you for the opportunity to present our data here. As you can take from our first slide, I'm going to talk about B-cell receptor repertoires in humans in health and disease. First, I would like to allow myself a short introduction on normal B-cell immunology before we turn to the lymphomas. Let's talk about germinal center reactions and B-cell diversity in first instance. This is supposed to be a healthy human, and a healthy human is full of naive B-cells. Please don't think they have different colors, but they have different antigen receptors with unique specificities. And upon an infection, you can see this human now turn red because he's infected. Part of these naive B cells may be selected via antigen contact, get activated home to the follicles, and then receive T cell interaction to undergo a germinal center reaction. During this very simplified scheme of a germinal center reaction, the naive B cell will be activated, receive T cell help, and then proliferate and introduce point mutations into the B cell receptor rearrangements, here shown as a red mark. Then, upon further cell divisions, further mutations will be introduced, and these here be unique, and the ones here in the beginning are shared. Also, part of the progeny may class switch, for example, to IgG, and then further rounds of proliferation, mutation, and selection will lead to the outcome of the germinal center reaction with a long-lived and high-affinity plasma cell secreting high-affinity antibodies to assess the invading pathogen. And also, class-switched memory B cells will be generated. They are long-lived, mutated, and of course class-switched, and they are prone to undergo secondary immune responses upon re-encounter of cognate antigen. Already more than 20 years ago, two further subsets of Ig-mutated B cells were detected in the peripheral blood of healthy humans, and these were called IgM-only B cells or IgM-IgD CT27 plus cells, and they showed clear mutated Ig levels relatively opposed to the class-switched B cells. And there was a lot of discussion about the generation or genesis of these cells, that they, for example, are generated in the spleen, or that they are also generated independently of germinal center reaction, which is still a big matter of debate, if you ask me. But today I want to highlight this point. They are very interesting in lymphomagenesis because far than 70% of human B-cell lymphomas carry Ig mutations, but are not class-switched and still IgM positive. And these are interestingly also often associated with characteristic infections like EBV, HIV, or Helicobacter pylori, or even malaria. So now we wanted to find out more about the clonal relation and the clonal expansions of these subsets. Especially, we want to understand if there are common generation pathways between these subsets, or also estimate clonal expansion as a matter of importance of an individual subset. So how did we do this? We sorted each of these subpopulations, and we used duplicate analysis. That's very important for the proper statistical evaluation. And then we performed VDJ gene deep sequencing, for example, with the Invivoscribe lymphotrack assay. So what we found practically was that among these memory B cells, a large fraction, about 55%, and we detected that in two independent donors covering about 250,000 cells per donor, of these sequences among these cells were unique. But we also detected clonal sequences that only consisted of IgG, only of IgM-derived sequences, or a small fraction of clones that contain both IgM and IgG sequences. But this was the interesting part because the larger the clones were that we looked at, we saw that the content or the fraction of the shared, that the mixed IgG and IgM containing clones significantly rised. That means the more sequences we found related to a certain clone, the more sure we could see that they consisted of both IgM and IgG sequences. And we concluded that the vast majority of human IgM memory B cells, is germinal center derived. So to sum this up, what does this mean? That means that already throughout the germinal center reaction, but already at very early stages, the different individual steps are remembered as long-lived memory B cells that we can detect in the peripheral blood even long after the germinal center reaction has finished. Now, these cells can be used, these memory B cells, to learn something about the dynamics of germinal center reactions. For example, we can sort the cells from the peripheral blood and then perform BCR repertoire deep sequencing, and we tend to use the Lymphotrack from Invivoscribe because it's just very efficient. And with these memory B cells and the BCR sequences and the mutational sequences, we can reconstruct the proliferation, mutation, and selection events during the germinal center reaction of these B cell clones. So this would, for example, then lead to this very simplified scheme of a genealogical analysis of a memory B cell clone. First of all, we have these different parameters. The B cell receptor gene rearrangement, of course, serves as a clonal marker, as a molecular fingerprint of the clone, if you want so. Second, we have the mutational sequence. Each node represents one additional sequence, and this may serve as a molecular clock. And of course, the shape of the tree would tell us something about the clonal dynamics, that is evolution, competition, or selection. Now, what we found was, for example, when we looked at the clones that only consisted of IgG memory B cells, that they have this, we call this giraffe neck-shaped structures. That means they have many shared mutations, and then here highly selected clonal members at the end of these trees. So they represent the high affinity subset of the clone. The IgM memory B cells, of course, had a much more bushy shape, so rather first primary diversification, and they are low affinity and flexible, so we think they rather represent the adaptive arm of B cell memory. They are open for further mutational selection and also for further class switching to independent subsets. But let me remind you, of course, the majority of clones that we detected from the larger clones was that these consist of both a highly selected part and a low affinity flexible part of B cell memory, and this was published. So now let's turn to chronic lymphocytic leukemia. Just short reminders, it's a monoclonal expansion of CD19 positive, CD5 plus, CD23 positive B cells, and they are shown here in red in this ill human. So the clinical outcome of CLL is highly variable, and there are diverse treatment options from watch and wait ranging to stem cell transplantation. So we can get rid of the majority of the cells, but unfortunately CLL is incurable, and relapse or progress often occurs together with subclonal competition. Now, also CLL development may take decades. There are several, so for example, very early mutations detectable long before clinical outgrowth, or we have the monoclonal B-cell lymphocytosis as a preclinical form of CLL. But the two things that interest me the most in CLL pathogenesis is, first of all, we don't know the cellular origin, so there must be the healthy, normal B cell that starts and then gets transformed to lead to a CLL disease. And second, the steps, the very early steps of the molecular pathogenesis are still rather unknown. Now, first our work on the cellular origin of CLL. This is a flow cytometric analysis of the blood of a 30-year-old healthy human, so he's definitely not a CLL patient, but he has in his peripheral blood CLL-like cells, CD19 plus, CD5 plus, CD23 plus, even sometimes CD27 double positive, exactly like CLL. So they show a phenotype highly reminiscent of CLL. And importantly, these mature CD5 positive B cells, as we call these cells, share typical CLL features as opposed to all the other normal B cell subset. This is a principal component analysis including splenic marginal zone B cells, class-switched memory B cells, IgM memory, IgM-only B cells, naive B cells, and here we have Ig mutated and Ig unmutated status CLL cases, and the CD5 mature cells shown here in orange, I hope you can see it, they cluster closely together in the transcriptional profiles and also stereotype receptor usage together with the CLL cells. So we concluded that the normal or mature CD5 positive B cell represents a very good candidate for the cellular origin of CLL. And then, together with Christoph Plass and Chris Oakes at the DKFZ in Heidelberg, we performed a very interesting analysis on the German center-dependent DNA methylation reprogramming. So remember, we have the German center reaction, and from each reaction step, we have these memory B cells putatively remembered in the peripheral blood. So what did we do? According to their molecular clock, we sorted these memory B cells and then performed whole genome bisulfite sequencing. And this led to this phyloepigenetic tree of German center reprogramming. You can imagine these are the along this axis you have the change of the different German center-derived reprogramming differences of the epigenetic patterns among these cells, ranging from naive B cells up to the highly class-switched and highly mutated high-affinity memory B cells. So when we added now the epigenetic profiles of 286 CLL cases, 450K methylation arrays, then we wanted to understand where are these located along this phyloepigenetic axis. And interestingly, they concentrated somewhere here between a lowly mutated up to the highly mutated class-switch memory B cell part. And all CLL cases, also those that were called as IG unmutated status, showed a German center-dependent DNA methylation reprogramming. And more importantly, as you can already get from the colors here, the intensity of the German center reprogramming determined the clinical outcome of the cases, and we published that in Nature Genetics in 2016. So this adding up to our scheme, we have the cellular origin, the normal CD5 positive cell, and for CLL pathogenesis, already very early, we very likely need the epigenetic reprogramming in a German center reaction to get to a CLL. Now we come to our recent findings on CLL sibling cells. Let me explain to you what this is. So each CLL is supposed to be German center experience and supposed to transform stepwise, of course. Now the point is, if at each of these transformation steps, memory B cells should be generated as occurring in every German center reaction, we should expect in the blood of CLL patients the so-called memory B cells, we call them sibling cells, which should be potentially normal or pre-malignant memory B cells that are corresponding to the CLL clone that breaks out afterwards. So indeed, we have to look at CLL patients in the peripheral blood. These are 3 different patients. And what you see here in blue are the CLL clones. But what we see here in magenta is the normal, we say, residual B cells. So in this patient, you have the CLL clone, but of course you have pushed aside the residual normal B cells. And they can stain them in with several markers, and you can clearly identify them. And then you can sort purify both of these populations and compare, for example, their clonality. And these normal B cells here in all of these 3 patients, they are completely polyclonal and they are immunocompetent in contrast to CLL. For example, they can very easily be convinced to secrete immunoglobulins. More importantly, we found that these cells, these normal B cells, also include surprisingly large number of CLL sibling cells. So how can I show you this? This is from these 3 patients, a CDR3 spectrotyping analysis, and you see that's are more or less polyclonal cells. There's a typical Gaussian distribution. And of course, the corresponding CLL clone is monoclonal. But what you see here at this point, that among these normal B cells, although sorted to high purity, you find the same rearrangement quite frequently associated also with the normal cells. Now what did we do? These putative sibling cells and CLL cells were sort purified, and we performed a lot of analysis. Today I will only talk about the B cell receptor repertoire signaling due to time. So this is a typical tree. It's also, again, very simplified to show you what we are dealing with. So in blue is shown all the normal B cells that we get from the residual cells that are clonally related to the CLL clone. So we have the common origin, we have the same molecular fingerprint. Now in orange, we see the cells that we only retrieve from the CLL clone, and in white those where we get normal and CLL cells at the same time. Of course, now mathematically, we can subdivide these cells into the CLL tree that only comes from the CLL subclone sequences and those from the sibling cells. And see again here, this corresponds to millions of CLL cells, whereas this corresponds to only a few hundred or 1,000 of sibling cells, because we cannot get much more cells out of this donor, but the tree looks completely different. And this is also mathematically can be proven by looking at different tree parameters like maximum in-betweenness, branching, intraclonal diversity, and other factors. And you see that they are all highly significantly different between the sibling cells and the CLL clonal rearrangements. So my first summary, in 32 CLL cases analyzed, we find residual B cells containing CLL sibling cells. And each sibling tree is different from the corresponding CLL tree, meaning that the sibling cells are not just sort contaminations, but display a higher diversity and also different selection features. And now again, coming back from these trees, we can again learn something about the dynamics of CLL pathogenesis. And it is that it is not linear as expected earlier, but that it is multicentric and occurs in parallel. You may have anticipated that the CLL cells are distributed throughout the whole tree, and the normal cells somehow surround this or embed the CLL cells. That means during CLL pathogenesis, and this is already long before clinical manifestation, CLL subclones are generated. And now, according to the aggressiveness of the CLL, the indolent CLL cases have subclones that largely consist, the tumor mass is formed by late germinal center excitants, but among the aggressive CLL cases, and we tested 20, so far could prove 20 from 24 cases, the tumor mass is located more early among the germinal center outcoming cells. So now we, of course, are interested in CLL relapsing and how the subclonality changes here. First of all, these are 3 indolent CLL cases. We have different time points sequenced deep into the repertoires. And what you see here is that the trees must shape because we have much more information here. What you see that when we sequence from all these 2-3 time points, that the tree shapes of this IG diversity among the CLL does not change significantly from the 2 or 3 time points of testing. So the subclonal composition in indolent CLL is stable. This changes, however, and we see here with this color coding, I can't show you the whole shape of the tree, unfortunately, because this would get too complex, you couldn't see it by eye anymore. So the sibling cells here show that the red-pink color code shows the most intense progress. So in year 2014, there is a change in the tree shape exactly at this position, whereas the other positions stay stable. And here we have 2 different time points, and we see that in this first year in 2012, there was a change in the clonality at this position, and 3 changes here in 2018 afterwards. So this is in progressing CLL cases. Now, let's focus on this first donor here. And this is the same CLL tree. And this is the same CLL tree as it's given by the CLL sequences. But here, the blue color code now indicates where the highest variation or differentiation of the corresponding sibling cells has taken part. And this dark blue means that the highest diversity between the 2 different time points that we analyzed taking place among the sibling cells was located exactly at the same position as here the progress suggested. And this is also true for the second patient. Unfortunately, we don't have cells, normal cells sorted from the first time point, but exactly here, where in 2018 from this patient, there was a clonal progress among the CLL cells, we find exactly at the same positions also the highest diversity occurring among the sibling cells. So this is very complicated to understand, so let me point you to this very simplified model. We have this one patient here, and he has a CLL progress, and then at treatment, the numbers of leukocytes and the peripheral blood drop, and then after 2 years, he has got a relapse. We sorted cells, CLL cells and sibling cells separately at these 3 different time points, and again performed with Invivoscribe lymphotract assay the repertoire analysis. This is the first time point, the CLL during the slow progress. So before treatment, we see this is an aggressive CLL clone because the tumor mass, shown here by the big bulb, here are the most sequences according to the CLL cells, are on the beginning of the tree. So we have early cells, and this is the typical clonal diversification and subclonal composition of the CLL. Now, after treatment, the tumor mass, of course, shrinks, and all subclones are below detection limit. But that doesn't mean they are not there, because all of them come back in the relapse. But what changes among the different tree is that the tumor mass shifted. So now the majority of sequences has shifted to these mutational subgroups. And importantly, look at these positions in this tree. They are exactly the same positions where the normal and CLL cells show the highest diversity. So it's not at the positions where we only have CLL cells. They seem to be not too much important for the progress, but it's there where the CLL cells and the sibling cells together show the highest diversification. So how pre-malignant are the CLL sibling cells? This is our current work now. Of course, we anticipate there must be some kind of pre-malignant subset. So we performed single-cell RNA sequencing, so this ongoing work. The idea in this is that from the CLL, we know the transforming events that we can detect there. Also, of course, we are very interested in epigenetic changes, of course. And then remember, we have the molecular clock of the clonally related cells. So now we think we can find the sequence of transforming events to understand the steps to the CLL pathogenesis before outbreak. That's our next goal.